I've worked in the area of neuroscience, doing that sabbatical leave with Erminio Costa, Video Georgetown Institute of Neurosciences, as mentioned before. And interestingly, during that time, I worked on things like phencyclidine, PCP, angel dust. And structurally, angel dust looks pretty much like ketamine. So there we were actually trying to make PCP antagonists, but many of you know what the properties of angel dust is. Also over the years, I worked on ergot alkaloids, developing new syntheses of ergot alkaloids like LSD. Worked on things like cocaine. We invented a compound called Nocaine, which could potentially be used to treat cocaine abuse. So a lot of different, sorts of projects that I've worked on over the years, and a lot of them have been fun. Many of these projects are inspired by natural products chemistry. Mother Nature is a great inventor, as you know. So psilocybin is, for a chemist, is a structurally interesting but structurally simple molecule, and it's related to serotonin structurally. Yeah. Which is, why, it has some of the similar effects, and it works at similar receptors, the 5-HT2A, 2B, and 2C receptors. And as Gideon and others have mentioned, and Ian himself, the psychedelic effects of a compound like psilocybin is due to its interaction, we believe, primarily with the 5-HT2A receptor. The 2B is the bad guy, because that's involved with the cardiac disease, and 2C is a good one. And interestingly, I got involved probably about 10 years ago with a very famous neuropharmacologist at University of North Carolina, Brian Roth, and his name has already been mentioned. And later on, you'll hear from John McCorby, who is a postdoc with Brian Roth. And John McCorby is also involved in Bright Minds Company. He's also a fabulous neuropharmacologist. But Brian and I were working on trying to identify new types of 5-HT2C ligands, primarily for application to schizophrenia, because the 5-HT2C receptor can actually control the release of dopamine in the mesolimbic system. So we thought that 2C ligands might provide a new type of anti-schizophrenic agent that would not have issues with weight gain, which a lot of these antipsychotics have, and would also not have an impact on cognition, because actually the 2C agonists can improve upon cognition, as shown by some experiments using NMDA antagonists. So eventually, after many types of manipulations that began with high throughput screening, where Brian took a library of compounds trying to find new types of chemical matter that would impact the 2C system, we came up with tranocipramine, which is a MAO inhibitor. We then manipulated that by making probably several hundred new compounds, and came up with some very selective 5-HT2C ligands that don't have 5-HT2B activity. And that led to this patent that Ian has already referred to, that's now been licensed from the University of Illinois Chicago to Bright Minds Biosciences. So that patent provides really at the basis for creating new types of compounds that won't be troubled by the 2B activity, but some of those compounds do retain some 2A activity. So we think we have already invented new agents related in part to psilocybin where we have factored out the 2B activity but kept the 2A and 2C. Some of these compounds have gone through extensive animal studies. We know they're safe, and now we're just selecting some of them for further studies that retain some of the 2A activity. And we'll be looking at these in various pain models, actually using one of the free programs available at NIH. So we think we have a leg up here in terms of positioning Bright Minds for moving quickly into the clinic.